use for this assignment. So we can hypothesize the effect of function changes on certain primary amino acids and then see how that changes the function of a protein. This is something I'll highlight with the video today. Secondly, we can model the potential structure of a protein based on other known protein structures, and then look to see how alternative splicing can potentially change that. So effectively, in this assignment, we're looking to see how splice variants change the function of a protein. We're always told from day one that splice variants allow for differences in protein. This will actually be the sort of physical proof of that with DeepView. And thirdly, we can look at conserved amino acid sequences within proteins to see why they are conserved. What is it that makes a conserved region in a protein conserved with regards to either structure or function? So I'm going to start to give a demo of the first one here, looking to see how primary changes in an amino acid sequence lead to structural and then functional changes in a protein. And then I'll do some spin-offs afterwards for basically options 2 and 3. So the particular protein I'm going to show or use today as an example, I'm actually going to do 2, so I'm kind of doubling up. You guys really only need to work on 1, but I'm going to look at 2 in particular because there's a bit of a contrast that I'd like to draw here. So my 2 proteins of interest are the retinoid X receptor, the RXR, and the retinoic acid receptor, the RAR. These are transcription factors which are activated by different isomers of retinoic acid. There's 2 main isomers. There's an all-trans or trans, same thing, or AT. These all sort of synonymous. But there's this all-trans retinoic acid, so it's very long. It's actually very similar to retinal that we see within the opsins. And then the other isomer that we have is 9-cis-retinoic acid, so it's kinked. Now, importantly, just like with the opsins, the 9-cis-retinoic acid is very short. If we kind of look at it with regards to length this way, it's kind of short and stubby, whereas all-trans retinoic acid is quite long and narrow. The RXR, it's not shown in this figure, but the RXR can be somewhat promiscuous in its binding. It can bind to both 9-cis-RA as well as all-trans RA, whereas the whereas the RAR is very highly specific. It can only bind to all trans RA. It cannot bind to 9-cis RA. So there's sort of a bit of a concern there. Not concern, but a stringency there that we don't see necessarily with the RXR. So this is something that I'd like to actually see. What is it about the structure of one protein over the other that allows for more promiscuous binding compared to the other protein? And then secondly, can I make a, say, highly specific protein more promiscuous? So can I make an RAR more like an RXR, and then it can bind to both of these isomers? Or vice versa, what would I have to do to the RXR to change its structure such that it would function like an RAR and only be specific for one type of isomer? So I'm looking to see the structural relationship, and that structure should give me the reasoning for their particular in vivo functioning. And then alternatively, I can then start to change that structure to see how I can then change the functioning. So that's sort of the overall goal here. And again, I'm doing part 1 of this, not part 1, I'm doing option 1 within this assignment. So I've gone to RCSB here. Apologies for the small screen, but I've gone to, let me make it bigger. I've gone to RCSB. I've just typed in retinoic acid receptor, and I get a huge list of different proteins here. Now, importantly, I'm trying to show that the ligand, so the binding of retinoic acid to these receptors, is a bit of a is my focus. So I'm going to have to go through this list of molecules here and actually look for something that's actually bound with what I want. So here I can see, for example, that we've got the RXR. It's complexed with some other type of acid that's not retinoic acid, so that's a bit of a problem. This wouldn't really work for me. It's also bound to some other coactivator, so this wouldn't work for me, even though it's a crystal structure, it's not the most useful. Here I've got the ligand binding domain of the RXR in a complex with, again, a different isomer, or a different molecule. Here I've got ligand binding of the human retinoic acid, so the RAR, bound to all trans. So that's great, but for my purposes, I want to show both the RAR and RXR. So the point I'm trying to make here is that there's a huge amount of information available, and you have to sort of be picky with what you're showing. Some proteins are fragmented, so if you're working on protein A and you're interested in how it binds to DNA or it binds to, say, some ligand, it's very likely that you won't find a PDB file of the entire protein that you're looking at. Instead, that protein, for crystallization purposes, is often broken up into fragments. So you might only get certain domains, and we see that here, right? Ligand binding domain. This isn't the entire RAR. This is just part of that protein that actually binds to its ligand, and you'll see that very frequently. Another thing you'll see often quite frequently is that instead of using the natural ligands, so in this case we're looking at retinoic acids, this all trans and 9-cis business, often there'll be synthetic agonists or antagonists or some sort of chemical of a name, sort of a serial number. And the idea there is that sometimes we're interested, or the people using PDB are interested in drug interactions. So they'll have some novel molecule that's been made and they want to see if it binds with a protein of interest. So often they don't necessarily show the natural ligand. Very often the natural ligand is very sensitive. It can either be broken down, or at least in the case here for the RARs, photoisomerized, things like that. So you do have to spend a fair amount of time poking through the RCSB files to actually get your protein of interest, which is partly why, again, I'm having people post a protein claim, which has a very short proposal as to sort of a breakdown of what you're doing. And hopefully by then you've actually looked to see the PDB file that's there and make sure it's one that's usable for you. If you have to change later on, that's fine, but ideally you're sort of being a little bit proactive in that once you've claimed a protein, you've hopefully gone on RCSB to find a very useful copy for you, or a very useful PDB file for you. Now, luckily for me, the RXRs and the RARs are very well studied. So if we take a look here at my first hit, crystal structure of heterodynamic complex of RAR and RXR, and specifically their ligand binding domains. So that's exactly what I want. It's got both proteins dimerized together, and it's showing the ligand binding domains of both of them. So this is beautiful. This is everything I want. If I click on that and then go to download files, I can get the PDB file. You can use either PDB file that's offered. I prefer the text ones. Usually this gives a little bit less problems with compatibility, but by all means, try either or. But effectively, I'm going to download that and then show it in PDB. And as I've already kind of queued this up, I have it here. Oops. Come on. There we go. Now, often you'll get an error message like this. So I've already downloaded and opened Swiss PDB Viewer. So if we go here, I should have probably started with this. So I've already, I believe, shown this at some point in the class, or at least made reference. We're going to go to Swiss PDB Viewer. We're going to download it. It's a zip file. Sometimes it works on Macs, sometimes it doesn't. So definitely, if you're a Mac owner, do start to look into this sooner, or see if you can perform this project on another computer, either say the Brock Commons or somewhere else. But you do need to use this program. It's freeware. I'm not trying to take your money, but we do need to use this program. So I've downloaded it and opened it. And often what will happen when you open a file is that you'll get something like this. Because these are crystallized proteins and they're fragments, we've had to do everything that we've seen in the protein visualization lectures. So we've taken a model with a certain electron cloud density, and we've tried to pick, or well, the authors have tried to fit an atomic model to that. And often everything is not a perfect 100% match fit. And this is just simply saying that there's some changes or some mistakes or some unaccounted issue with regards to their particular atomic model. So here again, I've opened up someone else's prediction of a model, and it's sort of just being upfront and honest with regards to any issues. So in this case, it's saying some amino acid atoms are missing. And for our purposes, that's fine. We're not going to be super detailed on this. We're not trying to publish in Nature. We're just trying to get a crude understanding of structure and function. You might have a longer list of errors. Sometimes an error window comes up. So when I click this, I get this big error window, a lot of red text, and it's basically just telling me what's missing. So for our purposes, we can ignore most of this. Now, here's the protein by itself. Looks like a mess. So our Swiss PDB viewer, where's the one I want? I'm going to walk us through how to use this actual file, or this program and this file. So by dragging this into the window, I get a big mess. This doesn't look like anything useful for us, especially to the untrained eye. But what hopefully opens is, in addition to this sort of window here, you also have a control panel. If not, you go to this window and hit, whoops, window and hit Control Panel, and this should show up. This little column and these buttons along the sort of the middle here of this window are going to be your new best friends for most of this course, or most of this assignment. So right off the bat, we have the 3D crystal structure of our protein. Now this actually just shows the atomic model. It doesn't actually give us the ribbon structure. So what we need to do now is first off change the visualization so we can actually get an idea for what's going on. If we look to the control panel, we see that things are grouped into columns. We have A, and as we go down, this is every amino acid that's present. So A, and then clearly we switch to B, and C, D, E, F, all the way down. These are different protein fragments. So protein A, or fragment A rather, is all indicated here with an A. If we go on to a different fragment that's in this crystal, that would be B, and then C, and D, and so on and so forth. If we look to fragment A, we see the first residue is alanine, and specifically it's residue 231 in the protein. Then we have other amino acids all the way down, but we can see that our protein starts at residue 231 all the way down to wherever it ends off at the end of A. So it's simply just a section of the protein that was found on our CSP, which makes sense because we've got a ligand binding domain, so that's great. But this tells us specific residues. And again, later on, once you start to use the tools, you can select different residues here with regards to the red text and do different things to them. The first arrow, or the first column after that, actually, sorry, I apologize, I kind of missed a column here. So we've got A, and then we've got the amino acids. Then we have sort of a little tiny thing in between. H is for helix. So you can see here, all of these would represent a helix, and we'll come back to that after. But if you see, there's H's and S's, basically for helices and sheets. Something without an annotation here is just simply a loop region between either a helix or a sheet. And then next, if we start to go over to the next column here, these V's, these are meant to be little check marks. So this is just showing the backbone. So alanine, I'm showing the backbone. All the other ones showing the backbone. If I kind of unclick these by left clicking, they all disappear. If I only wanted to show one or two, just the backbones, so the polypeptide backbone, I can show that. And I can either click and select one or two, I can left click and either select all or none. But let's say here we go to this helix, I'm going to just click off just the helices. And if we zoom in, so here's sort of a good button here, this button to the far left, that's a zoom in, so it centers and zooms in on your image. Here's a helix, and importantly, this is just the polypeptide backbone of the helix. There's no R groups yet. And I can click and hold down, as long as this window is selected here, to actually visualize this and spin it around in 3D. Really kind of cool stuff. This again centers and enlarges to the best of the screen size. If I wanted to zoom out, I would use this button here, the hand button, and I would click and hold. I'm sorry, this moves side to side. Zoom out to the next one. I can also move up and down. If I wanted to zoom out, it would be this middle button here. I can zoom out and then zoom in. And again, I can always center if I don't like what I'm doing. And then again, I've got the kind of spin around in 3D option as well. So I'm showing just a helix here and just a backbone because that's all I've indicated here with this particular first column with regards to the ticks. The next column shows the actual R groups. So if I take a look, proline, proline's bent. You can actually see that right here. And valine, threonine, all the way down the line. Clearly I missed one there. But that's the actual helix now, with all the R groups jutting out from it. If I didn't want to show the R groups, I can simply remove them and have a backbone. If I wanted to label certain key residues, say, say my proline and my lysine, I can use this next column to actually show those labels, proline and lysine. No matter how I spin it, or even if I include the entire protein now, I can see where my proline and my lysine are, right here and right here. The next column shows electron cloud densities. We already saw this with the regards to the opsins. So if I do that and that, I can see the electron clouds that are produced. So you basically can't have overlap of electron clouds. It's not a good thing. And then lastly, which is probably more important for everybody, is that you can have ribbon structures. So here, I can highlight all of these and actually show a helix, which is much more interesting for us. So if I showed the entire protein, just the backbone, this isn't the most sort of clear, right? With all the residues, it's even more unclear. This is a mess. So by taking off the residues, just showing the backbone, at least initially, and then showing the ribbon, you get a lot more cool structure. You can even just remove the backbone entirely and show just the structure of the protein. This little drop-down arrow here, this black triangle, you can change certain things. So you can look at van der Waals forces, other accessible areas, and again, more tools for you to play around with. Lastly, we can color code things. So if I wanted to color code this helix, just the helix I was playing with, we can select all of them, say make them red, and I can see where I am within my entire protein. Oh, I apologize. There we go. So there's my helix right here. That's what I was playing with earlier. Now, importantly, you would have seen I used this drop-down arrow here. I can color the backbone and side chains. I can color just the backbone, the side chain. I can do the ribbon. I can color the labels. I can color a number of things. So if you're wanting to do the ribbon, you're going to have to select ribbon. The default is this backbone. But this sort of gives me everything I want, at least to start with. So we know to zoom in and center. Sorry, we know to center, we know to move left and right or up and down, wherever we want. We know to zoom in, zoom out, and we know to rotate in 3D. So that's a great start. Now, importantly, this protein, and most likely yours, is either going to be, is most likely rather, going to be a mirror image. So crystallization is often done with multiple arrangements of proteins, so this isn't a mirror image. So ideally, I don't want all of these. I'm probably only going to want to play with my A and B. So if I go down and select just for A and B, there we go. If you take a look, whoops, I'm going to show not only the backbone, but I want to show the ribbon as well. We should have A and B here, and you can start to see that. I haven't gotten all of B. Clearly there's some missing bits here. If you go down my list, I just kind of crudely stopped partway through, but if I kept going, you'd see I'm getting more and more of this B here. And I'd go to the end. But clearly I've got A over here, B over here, and now I can start to work on it. Another important thing is if we go to the very bottom of this list, if you're working with ligands, you don't have to be, but if you look, some of these don't make any sense. These REA, this isn't any amino acid. This is a ligand. So if you're working with a ligand or other, like say a DNA molecule, there might be an option for it to be shown down here. To help to highlight these, I might make these, let's say, neon green. And if we take a look to see where they are, you can see where they are. So here's one in here, another one's over here, and these other two that I'm showing, these are in part of that the mirror image. So again, this original PDB, it was a mirror image of a dimer. So there's another one, another protein here, and I'm removing them. It's because I'm just going to work with one dimer to make it simpler. But you can see one residue, or sorry, one ligand here, one ligand here. So that's a quick crash course so far through just this protein. Now, with regards to my original purpose, find the RXR and RAR and explain why they're hetero or why they're promiscuous versus stringent binding, I'm going to have to do some research, see where the ligands are, and I can kind of see that here, and then I could zoom in and actually start to work on this, either with primary research or other tools or techniques. But if I wanted to see where the ligands are, with regards to where they're binding, the best bet would be to go to the primary research and actually read about it. But here I'm just going to quickly color code these, so I can see a red molecule. And let's say I'm going to work with, let's do this guy here. I can physically zoom in. And again, this gets a bit messy, which is why it's always better to do primary reading. I mean, if you are just clicking blindly, you might not get every residue in the region that might interact with this. You definitely do need references for this paper, so you can't just be clicking away. You do need some type of justification to talk about the residues you're looking at. But as you can clearly see, by showing just the backbone, if I had everything, it's a mess, right? If I just show the backbone, I can start to click to see what's in and around the region of this ligand. So by using this question mark here, I can click the backbone and actually look to see what's actually here. And actually, if you look to the red text here, as I move the mouse over, it changes, right? So phenylalanine 295, over here glycine 294. Alternatively, you can click. And as you see, it puts a yellow label on it. So if I tilt that so the text is a little clear, that should say serine, S-E-R. If I got rid of the ribbon, it was S-E-R 280. So you can either hover the mouse over with this question mark or click it. Problem is when you click it, that yellow label doesn't go away. So then you'll either have to save the file. So you can go to File, then Save, and you're going to want to save current layer as a modification, or just start from scratch again. But again, usually with primary literature, it'll tell you the residues, and that's good to go. So what I've done, I've gone in and I've color-coded a couple things here for us. So I've got the RAR in light blue and the RXR in dark blue. And that's sort of a good starting point. If you're looking at a project, you're going to have to give me an introduction to the proteins, kind of walk me through what the protein normally does. Before we change around anything for structure and function, we need to know what it does. So you need to give a quick introduction. This introduction shouldn't be a lit review. It shouldn't be a list of 30 pages of text. It should just be a very quick, my protein does this, here's how it mechanically can do that, and here's the structure-function relationship. You just simply want to establish a foundation on which you're then going to build. So it's not a review of what's been done, it's not a review of how the protein works, it's a quick intro as to what the basic premise is, and then from there, the bulk of your report is going to be how you can change the structure and relate that to a change in function. So again, you're undertaking a novel approach or a novel experiment. In a sense, you are experimenting. You're conducting research here. How could you change this protein to change the function and structure? You're not going to review other research and simply just tell me what's been done before. You do need to take an independent approach to this type of a research task. So I'm clearly showing this. I would probably take a screenshot at this point and then put that into my Word document so I can kind of walk through. I could then sort of, again, with research and some work with Deep View, I could select just the helices that are interesting for me. So in this case, again, on my control panel, I've only selected helices that are interacting with my retinoic acids. And if you take a look, I've just selected them here. And in this case, again, my overall goal was to work out why one molecule can bind to both isomers of RA versus the other molecule only binding to one. So I've taken a look, and just under Deep View, I can see that, well, there's some helices that seem to get in the way at times. So both of these retinoic acids that are shown here in red, they're kinked, right? This one's almost like a 90-degree down. This one's almost a 90-degree up. These are the 9-cis isomers. Again, 9-cis seems to work really well with one receptor and not the other. So we want to see why that's the case. And importantly, we want to see why all trans doesn't really work too well in some cases. So if we take a look at the dark helix here, the RXR, if we take a look, we can see here that if we could imagine, if you will, an all-trans isomer kind of going straight out, right? All trans, if you go back to my PowerPoint image, which I'll show you, all straight versus kinked. If we take a look, and maybe zoom in a bit, if you imagine that you replace this with an all-trans molecule, this tail, this hydrophobic tail, would have to continue outward. So I'm going to rotate this so that that tail would be coming out of the plane of the monitor. So if I do that, look what happens with the helix. The helix gets in the way. If I swing it around this way, so again, if you imagine that this tail would continue to move straight, it's going to kind of pierce into the plane of the monitor as I bring it around. So just be going straight through here. That helix is in the way. That's a problem. That provides a steric hindrance. It's just simply physically in the way. No angle we can look at here seems to indicate it's going to be able to move around. So all trans will not fit into this particular molecule here. The darker one's the RXR. So it will not fit very well. All trans doesn't seem to work there. But if we look at the coloration here, actually I apologize, this should be the other way around. Oh no, we're good. So if I look at this one here, this other one, the RAR is the light one, it can accommodate 9-cis because clearly it's in the pocket. But if we imagine, again, all trans going straight out, if we move, look how that, if it comes out of the plane of the monitor, that helix is out of the way. So it'd be kind of jutting straight out at you right now from here. This helix is tilted off to the side. If I show the rest of the protein, which I won't do because then I ruin what I'm just indicating here, but with the rest of the protein, oh, I just lost the ligands. With the rest of the protein, there's really nothing in the way. So one of the things that seems to be describing, or at least accommodating this particular shape, is the binding would be the orientation of these helices. This helix is in the way, so physically all trans-retinoic acid does not fit in here, versus this helix, 9-cis-RA fits as well as all trans. So this explains why this protein is promiscuous and combined to either, because it can physically allow it. Again, this is the tail. If it juts out, as I rotate, it should pierce into the plane of the monitor. It's free and clear. Even if I show all the residues rather than just the backbones, I just show, oh, it's the other one, go back up. Even if I show all the residues, there's nothing really too much in the way. It might look like from this angle, this residue's in the way, but if you look to see the overall angle here, it would be going this way. So it actually fits in between, right? So here's that kink, imagine it going straight out. So right where my mouse is, right here, in a way, if you almost put your finger on the monitor, as I rotate this down, right about this alignment, that should be coming straight out at you. So in a sense, it's actually sandwiched between these two residues. And importantly, this is a hydrophobic tail. These are hydrophobic residues. They all integrate very nicely. This helix does not allow that. There's a steric hindrance. There's a blocking of that R group, or sorry, there's a blocking just by the backbone of the helix itself. But if we were to continue, it just simply cannot fit. It gets in the way. Even though we might have a hydrophobic helix here, there's some charged ones over here, and that just simply gets in the way. So there's physical hindrance as well as sort of a charge distribution there. So I could indicate that with a few pictures, kind of show what happens normally. Oops. This I'm not going to show yet. Let me get rid of that. But let's go back to my overall protein right here. So again, I have my two helices. One can accommodate, the other one cannot. I can now start to investigate why, or well, how could I change this? So I'm going to show the entire protein. Oops. Sorry, give me 1 second. That did not work well. Let's go with this one. There we go. So I've got my entire protein. In this case, my ligands are colored, I believe, green. But I've indicated, and again, you'll be taking screenshots, and this report is essentially an essay report. So you'll be writing up a bunch of stuff. Actually, no, in a way, it's more of a lab. You'll be writing up an intro. You'll then write up a couple quick reports or results showing the basic default of how the protein works, and then you'll start to show an investigation. So at first, we then sought to determine whether we could change a protein, and then you'd show this. You'd show a couple pictures, and then you'd write things up. So how could we change this to skew one protein to behave more like the other? Well, really crudely, I could start to change the overall structure. If I look at this protein, here was the helix I was playing with before. This helix is kind of sideways. Now it's going into the plane of the monitor. I could change this in such a way that it might actually allow me to go in and maybe skew that helix to the left or the right. And an easy thing to do for that, give me 1 second here to just get rid of all the extra filler. I'm just the wrong way, C and D. There we go. So now I'm just showing the backbone. I'm showing some other fragments, that's fine. But if I can somehow make a mutation here to skew or bend one of these helices over, that would be great. I could swing a helix out of the way. I could swing a helix in the way. So let's take a look at this structure. And again, it's going to take some time for you to just kind of spin this around and visualize it. But I see that my helix is right here. I could potentially take a residue over here. I see this sort of loop region here. If you imagine that there's some smaller residues in this region, if I made a bulkier one, that would help to pinch this turn tighter. So imagine, actually might be easier to say here, right? These were all very flexible glycines. I could put a string of tryptophans on this side, and that would promote a turning of this region here to turn more tightly. So instead of being a very gentle arc, it'd be a very sharp arc. Same idea here. There's a turn here, little sort of region here. If I changed a couple of residues to very bulky ones, bigger than whatever they currently are, then I could promote a tighter turn. Conversely, if I took whatever residues were there and made them smaller, I could promote a more gentler turn. And by doing that, that would skew the entire shape of a protein. So if I take my turn here, and it's a little hard to see, but if I take this turn here, I can now start to change the angles and mutate things. So the first thing I would want to do is show the normal turn, maybe talk about an angle of a turn. So if I click different residues, and you have to actually click the atoms here, you can actually get different angles of alignments here. So if I click this, this gives me angles. So I go A to B to C, and I could actually get an angle between my 3 points. I could show a particular angle. In this case, I'll explain this double vision in a second. Whatever angle I have here, it's showing at 29.25 degrees. I could then change that angle to something else. What I would do is I would mutate a residue. So I'm going to, and I'll need to zoom in a bit. I'll explain that double vision in a second or two. Let's say again, I'm going to target this region here, and I want to change some stuff around to promote a tighter turn, because hopefully that's going to skew things to swing this helix here. Again, if I color coded it a little bit cleaner, you'd be able to see it better, but this helix here is what's going to physically block our all-trans retinoic acid, very similar to the image I showed with just the two helices. This is that helix that was shown in that previous file, and this is what's moved slightly out of the way such that this protein can accommodate both all-trans and 9-cis residues. So ideally, I'm going to hypothesize that a number of large mutations here, from small residues to larger ones, will actually cause a kinking of this particular strand. So I'm going to click a residue. When I do that, I get a list of things that come down. These are all the different residues. So I'm going to go to oh, it's loading for some reason. I'm going to go to a tryptophan. Oh no. I'll reload. Give me 1 second. So this is actually a really good opportunity to kind of tell you guys, you'll definitely need to save things as you go, because clearly things, anything for a computer definitely can be a problem here. So actually, let's keep it a little simpler, because it's better if I can show things a little bit more clearly. So let's go back to this. I'll keep it simple. This helix is bent. If we could bend this helix such that it swings over, we could have it so that it's not in the way. We could either continue the bend, or we could bend it back in a different direction. But clearly there's a bend here, and that bend is causing a physical blocking, right? This tail of the retinoic acid molecule cannot go into the helix. So I'm going to go to that section that I've indicated on the helix. It's the wrong one. It's my light blue helix. I want the dark blue helix. Here it is. Because I'm now working on a very small scale, I can have all the residues and everything in place. And again, this is in the way. So I could hypothesize, and I'm going just to get rid of this for a quick second, that actually it's easier if I show it. If I was to change, say, this residue to something big and bulky, that might promote a more steep turn to the side here, which might allow this helix to swing out of the way and allow the dark blue protein here to actually accommodate both residues. So if I mutate this, I click, I can make it say a tryptophan. And now you can see there's a tryptophan. Importantly, when I click that, there's little black arrows here. I can move these in different orientations. This shows all the different conformations it can have. Now, importantly, if you look, there's a pink dashed line there. That shows a steric hindrance. That shows that this particular orientation is not allowed. If I had this here, it would block the retinoic acid molecule and actually make this protein entirely dysfunctional. It wouldn't respond to any ligand because you're now physically blocking retinoic acid. So certainly, at least with my context, where I'm trying to make it bind to more retinoic acid, I wouldn't want that. But if I was trying to mutate this protein to make it non-functional, that would be great. This is an entirely open-ended assignment, so you can do what you want. You can make a protein better, you can make it worse. But I'm just going to pick this conformation. I'm going to click anywhere, and then click mutate. It says, do I want to change this? Leucine to tryptophan. Well, yes. I've now made a very big and bulky residue change. Presumably, that's going to change the overall backbone here. If I click torsion, I could sort of hypothesize what that backbone change would do. So I click this, and now I get a bunch of black arrows here. Depending what I click, I can rotate this helix into different orientations. So these are a little tricky to click, but if you can get them, I can then rotate these around very nicely. Oh, that's quite quick. See, so I'm obviously not going to go crazy with it like that, but if my original protein was like this, I could go in and just very gently, just tweak it just a little bit. I've only made one residue change, so you're not making a huge impact, but I could tweak it, just say like that much, and then maybe it bends that much. I hope everyone saw that. I'll do it again just to exaggerate, but maybe not. There we go. Down a bit, kind of out of the way. And it says, do I want to accept this? I'll go yes. So now let's take a look at what my tiny mutation did. It massively moved this, so I've sort of exaggerated greatly. But if you look now, if this was an all-trans molecule, you'd have a tail going straight up and down. And if you look, it's free and clear. There's nothing blocking it now. I've taken this stringent protein and made it more promiscuous. And obviously I've done this here to an extreme, so it's probably not likely to happen. But that's just an example. I could mutate it a different way. Again, if I wanted, I could look at angles. I could do an angle analysis to say head to tail of this particular protein from a bend. But you can do almost anything. You can take an active site of a protein, make it charged. If it was uncharged, you can make it positive. If it was negative, if it was hydrophobic, if it was charged, you can make it uncharged. You can do almost anything. This is an entirely open-ended assignment. The idea is to think of this almost like Lego blocks. You can build almost anything you want. Now, the only thing to this, or the only challenge, is that you have to justify your position. So I've proposed a massive change in the overall structure of this protein. If I zoom out, it's quite dramatic. I've changed the overall protein. If I was to kind of show that again, I could see if I can click. It's a little tricky. But if I start to move this, look how the entire protein changes. You see that? Quite dramatic. So I can't propose anything under the sun, because if I did that, I'm probably going to have steric hindrance. If you look, this helix has completely collided with this protein. This loop region is physically overlapping with another helix here. Deep view does not accommodate or account for the presence of other atoms here. So in a sense, you've got two atoms or two parts of a protein that are overlapping. So I have to justify that this is a likely conformation to go into. The way to do that would be to look at folding as one potential mechanism, right? So I can actually go in, tools, and tools gives me a huge bunch of everything a huge bunch of analytical tools. I can look at electrostatic potential. I can look at the surface charge of this protein. One of the common things is to compute energy. And it's usually, I believe it was force field. So this is calculating the overall energy. Sometimes takes a while because there's a lot of interactions. I don't really care about any of these details as I go down. What I do care about are the things in red. So this is showing there's alanine from whatever residue at whatever position. It has a massive delta G value. So this last column is the energy of folding, so of this specific residue. And if you look, you can have slightly positive, you can have slightly negative numbers, that's fine. Generally for folding, it's just the same as thermodynamics, you want a negative energy value. Some of these are ridiculously positive. This is almost positive, probably about 1 billion. So that means this is not a good energetic state for this protein to be in. At the end of the day, we're interested specifically in the total. So our E-value here is a ridiculously positive number, which means this will not fold spontaneously. So in other words, the mutation I've made with that specific twist or turn is not going to happen in nature because it's so ridiculously positive. For anything to be thermodynamically favorable, it has to have a negative delta G. Another way we could also look at this, so in that case, what I would do is I would go back and mutate this to something a little less dramatic. That was kind of way over the top. But if I go back to my unchanged protein, I could go back and change it again. And again, I'd probably have a slightly lesser dramatic turn. But what I can do first is calculate the energy, sort of the wild type. I don't think it went. Doesn't seem to want to go now. Come on. So this is going to calculate, and just to show there's sort of nothing up my sleeves, this should give us a very negative number somewhere. If you look at E total is negative 11,000. If I make a mutation, so now I could write that number down, give the reader sort of a starting point, but here's a mutation, I'm going to change the torsion, so I'll pretend I've already done the mutation. Just going to click, try to get it. I'm going to have to zoom in. Torsion, click. So now I'm just going to do a very, very mild motion. So just a fraction. Go ahead and do it. Come on. Clearly this is a little bit tricky. Definitely need a mouse if you've got a laptop. But here we go. Did you see that? Just a slight, over here will change, just a fraction of a millimeter. Might be enough to get me that clearance I need for my ligand, but I'm going to click and see, just a touch. Going to accept that. And I'm going to calculate my energy again. I started with negative about 11,000. This is going to go, hopefully. If I take a look at the bottom now, it's about positive 13,000. So I kind of messed that up a bit. So clearly I've made too much of a change. It'll depend where your protein is, right? So I've used this one helix here underneath. If I can zoom out a bit, it's probably collided with the helix above it. Or you can at least follow it and see where it's collided with. So the motions you're making are going to have to be very minimal. You can't sort of be all crazy. This is building, I'm trying to tell you on the building block idea, but different regions of the protein are going to be very either accommodating or non-accommodating of change. For example, if I change this loop region along the top here, where there's nothing really in the way, this whole loop here, that could swing out over here anywhere I'd want, and it'd probably still have a negative energy for folding. If that swings inside, well, it's going to collide with other molecules and not be as useful. The idea is that I have to provide justification for the changes I've made. So I can propose any mutation I want, that's fine, but then the resulting structural change and the functional change has to be somehow justified. I can show, if I'm looking at ligand binding, I can show the presence of hydrogen bonds, the absence. I can show overall alignments or spacing. So if I click, say, this button here, I can do spacing. In the same way I could look at angles, I can click two atoms and give the space. So here, those are 13.38 angstroms apart. I could make an active site that's normally wide, very narrow, or a narrow one, very wide, vice versa. I can do energy of folding. Now, importantly for folding, because we haven't covered it yet, as long as folding is negative, everything's happy. So you don't have to have an increase in the negative value to promote or justify your mutation. If you start with something that's negative 1,000, as long as it stays as -1 or more, you're fine. So -1, -2, -3, those are all fine. The fact you've lost energy doesn't matter. The issue is at the end of the day, there has to be a total of a negative number. Any negative number is fine. And again, if you make a mistake, that report tells you where things are colliding. So you might be able to tweak things a little bit more differently if you look to see where those residues are. You don't have to do folding. You can do a whole bunch of other things. It all depends on what you're doing when you're approaching this question. If you're looking to change an active site to make a protein more stringent or less stringent, you're either going to dramatically change the charge or the size of that area, in which case you could then look to see sort of overall surface charge. There's a whole bunch of different things here that we can look at. And again, we can calculate surface charge, we can look at different fits. The idea is that you spend some time going through some of this here. But there's a number of different tools you can do. And again, look at different hydrogen bonds, look at different distances. At the end of the day, you're making a mutation. You're looking to see how that changes the structure, and then you're going to hypothesize that change. You'll then back it up with some hard data, some either numbers or display of bonds, display of surface charge, display of topology, energy of folding, things like that. And that's your proof that your hypothesis would be correct. I'm going to change a protein. I'm going to make this mutation. In my case here, I'm making a stringent protein more promiscuous, or vice versa, a promiscuous protein more stringent. And I'm going to prove that, one, by showing the physical overlay between my ligand and the protein. And then secondly, whatever structural change I've done, I'm either going to prove with folding or prove with something else. And that's really about it. Really sort of long-winded, but that covers all of part one. Part two, very, very similar. We're looking at different splice variants. So all I would do is say, let's pretend for my hypothesis here that I've got a particular variant. Come on. I've got a protein. It's known to have a mutation that deletes, say, alanine 207 through to isoleucine 230. I could remove that section from the protein. If you take a look to see where it is, to make it easier, I'll color the ribbon. Let's say red. And let me show the entirety of this protein. So I just want to show B, because that's part of my protein here. B, go all the way down. So I'm just showing my protein B. There's a known splice variant in the literature, and I'm trying to ask, well, what does that splice variant do? And again, because this is something that binds to a ligand, it might be useful to show them. I'll even color code them. Color code the backbone. Make these, let's call them green. There's my ligand, right there, blinking. So remove everything else, center it. Oh, I'll explain that double vision near the end. Almost done though. So clearly, the red helix here is my, again, I got a hypothetical splice variant, seems to lose this helix. That helix seems to play a role in the active site. So if there's a splice variant that loses this, because there's a loop region here, presumably this loop region just bends and links up over here. So if it's just a deletion, we just simply remove this helix. In which case, that particular variant probably has a compromised function. If there's no active site or a damaged active site, this protein might be non-functional or what we call aberrant. In which case, that variant doesn't seem to cause any function. You're basically shutting down the retinoic acid receptor. Other variants might add a different loop region. Perhaps that loop region, if you run it through PSORT or run it through another sequence analysis like what we've been covering, it might be a localization signal, in which case that splice variant changes its location within a cell. If there's a loop region or a helix, it might be, and it's hydrophobic, it might be a variant that allows that protein to be inserted into a membrane if it normally isn't. The idea is you can hypothesize the functional change in the protein due to the structural change that's seen in the two variants. So you'd get a wild type protein, you get your variant, and you'd color code the differences and look to see, well, what the differences are. Has the active site stayed the same? Can it still bind to DNA? There's a structural change, and you would start to work out what the functional change is. And then lastly, for option 3 here, analysis of conserved amino acids. So this is more of a report. But essentially what you're going to do, let's say, again, we're going to stick with retinoic acid receptors. I want to know what's the most crucial component of a retinoic acid receptor. So what I'm going to do is I'm going to go to NCBI. I'm going to search for protein, type in retinoic acid receptor, and I can get all sorts of protein sequences of this retinoic acid receptor. So here I get all the different organisms, right? So here's zebrafish, here's the newt. Some of these I actually, to be honest, don't know their species. When you click, they tell you. If I click on this, once you load it up, it tells you what it's from, the Pacific white shrimp, and I can actually go off and get the proper amino acid sequence. I would do this for about a dozen or so different organisms. You can use a number of different number of different programs for this, but I'm using one called BioEdit. This is another freeware program. You don't have to use it. There's dozens of other ones. But I've gone ahead and already gotten a whole bunch of different organisms, RXRs, that are shown here. I can then run an alignment. So I go to accessory program, run Clustal. This is a type of an alignment, and we don't need to know how it works. We're just going to run it. We're going to run it. By running it, it gives me now an alignment. What BioEdit allows me to do is actually visualize all the homologous regions. So I can search through this, and clearly because I've got a wide range here, right? I go from mammals all the way down to, say, crabs, and gastropods are like snails and slugs. So I kind of have the entire animal kingdom here quickly represented. By changing the different display properties here, I can actually get different sort of representations. This particular one here shows what's common across everything. So if you look, there's a high degree of variation on the front half of the protein, high degree of variation near the back half of the protein, but anything with columns like this are very highly conserved. So if I take a look, say just go right here, really highly conserved section. And if we look to see some of these other ones here, we see that if we remove lamprey and nematode, we'd get even more. So if I delete these two, delete sequences, and nematode, you get a big chunk here of very, very similar. Actually, even go ahead and get rid of sponge. You get a big chunk here of highly conserved residues. And if we wanted to see just the absolute core of the protein, so if you could boil it down to the most important 3 or 4 residues, that's fine. So you would have to actually have even more sequences here to see what's in common. But if you want to see a chunk of a protein, you'd use fewer animals that are all closely related. If you want to see the bare bones essentials, then you'd have a wide range of species here. But you would simply run an alignment and see what's common. And from this, I can see the residues. So I can see, and hopefully from one of these, I'd get the very specific PDB file. So you'd want to make sure on NCBI you can get the PDB file from this. Let's say I had the one for newt. I can go in and say, okay, well, residues, in this case, actually, let's not use newt. Let's say gastropod. Residues 120 through to 145 are incredibly important. I could highlight those on the PDB file and actually see what they do. And then from here, I can make reports showing why these are conserved. They're conserved because they're either, we'll say they maintain a certain curvature of the protein to allow the structure to be correct. They might be conserved because they're the active site residues and they interact with the ligand. It all depends on your protein. So overall, this is a very open-ended assignment. And you can either take a protein, make it better, take a protein, make it worse, make it more promiscuous, less promiscuous, functional, dysfunctional, membrane bound. You can do almost anything. The key is that your statements have to be supported by some type of analysis. And for that analysis, we either use the BioEdit or DeepView. Most of the time, we'll be using DeepView quite heavily. If you're going to propose a change, it has to be justified. You can propose any mutation. It's just a random fluke in evolution. That's fine. But you have to propose, or you have to provide justification for your hypothesized results. So here's a mutation, happens at random, that's fine. No justification needed. But if the helix swings out because it's a bulky residue, that's sort of justified. Bulky residue probably puts torsional strain on the backbone, therefore it's justified. To prove that the specific change in structure that you've now shown in DeepView, so say you've twisted a helix or you've now changed the distance in an active site, that can be supported by analysis. Again, folding energy, looking at distances between key residues, looking at the charge distribution or the electron cloud coloration for positive, negative charge. All those are hard data you can use to support your statements. So I hope that makes sense for now. Any questions, please do email me, but that's about it with regards to the assignment.